Good afternoon. I don't know if this is now switched on. Okay. Good afternoon and welcome to the Oxford Martin School. We have a slight change of plan to the advertised speakers because only one, Chaz Bountra, will be speaking initially, and then we'll have question and answer, which will include Lee and other colleagues. So I will introduce Chaz now. Chaz is co-director of the Oxford Martin Programme on Affordable Medicines. He is also Professor of Translational Medicine in the Nuffield Department of Clinical Medicine here, Chief Scientist at the Structural Genomics Consortium, and Visiting Professor in Neuroscience and Mental Health at Imperial. He has experience of drug discovery from the commercial side as well, as he was previously Vice President and Head of Biology at GlaxoSmithKline. As I said, Chaz will speak. He'll then be joined by his fellow directors on the Programme on Affordable Medicine up here to take any questions, along with Colin Bennett, the visiting fellow on that programme. And I just want to warn you that for that interactive part of the session, we are webcasting and filming this. It's being broadcast, so people just be aware of that and only ask questions if you're comfortable being filmed. So without further ado, I'll hand over to Chaz.
Well, thank you very much, Julian, and ladies and gentlemen, thank you for coming and giving up your time to listen to what we're trying to do. So Julian gave me a title of Transforming Drug Discovery, and that's what we're trying to do. And he said pathway to innovation, and I'll talk a little bit about innovation. Now, before I kick off, I'd like to introduce my colleagues. So Colin, who's sitting here. Colin, do you want to stand up and say hello to everybody, etc.? So Colin is a wonderful guy. He lives in Stratford-upon-Avon, and he's been helping us with this project just for fun, because he feels this is important to do. The poor man doesn't get paid anything. So my colleague Lee, Lee and I work very closely together. And then there's Nick Scott-Ram, who looks after the Academic Health Science Centre here in the university, which is the university working closely with the hospital and local industry, et cetera, et cetera. And so these are some of our colleagues. We've also got Adrian Towser, who's based in London, a lady named Olga, who's currently in Aston, and another colleague, Javier Lozan, who unfortunately couldn't make this evening, et cetera. So, but these guys will help us with the questions at the end. Now, James Martin, of course, we all recognize is a real visionary. I mean, he wanted us to think about global challenges and how we can address global challenges by bringing together multi-stakeholders, multi-disciplines to try and solve these problems. Now, we believe that trying to come up with novel medicines, effective medicines, which are affordable and doing it quickly is a massive global challenge. It's currently a challenge for patients, partly because we just don't have enough new drugs, and there are many diseases out there where we have no drugs. It's a challenge for the economy. I mean, the NHS, as we know, is facing serious challenges because they don't have enough budget, etc. It's also a major scientific challenge, and it's also an organizational challenge. So I think this absolutely fits James Martin's vision. A global challenge where we need to come up with solutions, and the solutions are bringing together people from different areas, different disciplines, different organizations, different backgrounds, etc. So what I'll try and do in the next 40 minutes or so, I'll split the presentation into 3 bits. First of all, I'll paint a picture of what I see as the problem, what I see as the challenge. And I'll go into this in fair detail, and I hope it isn't too gloomy. I'll then share with you what we've been doing in Oxford for the past decade. And this may be a solution or a partial solution. And then I'll finish off with what we're going to try and do with the help and the support of the Oxford Martin School. So let's think about the problem. Let's think about, what do patients want? What do patients' carers want? What do their relatives want? And what does society want? They desperately want novel drugs. They want drugs that are effective. They want drugs that are affordable. And they want them quickly. Let me just throw a few numbers. Cancer. In the UK, in the next 12 months, 350,000 people will get diagnosed with cancer. That is 1,000 people every day. That is one person every 90 seconds. In the next 60 minutes, 40 people will get diagnosed with cancer in the UK. Half of us in this room during our lifetime will have a diagnosis of cancer. In the next 12 months, 14 million people on this planet will get diagnosed with cancer. We desperately need new treatments. I worry a lot about dementia. In the UK, today, we have 850,000 people with dementia. In 2050, we will have 2.1 million people with dementia. 2 cities the size of Birmingham with dementia. All of us who live to more than 80, 1 in 6 of us will have dementia. The average cost today of caring for a dementia patient is £32,000 a year. And that's despite the fact that 2/3 of the costs are borne by the carers, so not by the state, not by the government. £32,000 a year. If you add all that up, today we are spending £26 billion a year looking after our dementia patients. Incidentally, 26 billion is, if you add up all of our household energy bills in the UK, that is 26 billion. That's how much we're spending looking after our dementia patients. And since last year, we now have more patients than we have carers. And we've not managed to come up with a new drug for dementia since 2002, 16 years ago. Mental health, massive crisis. You know, 1/4 of people will have some sort of mental health challenge during their lifetime. 83 million people across Europe have some sort of mental health issue. 20% of adults will have some form of depression throughout their lifetime. I was staggered to learn recently that if you look at 15-year-old girls, 1 in 150 will have anorexia. Imagine a normal secondary school of about 700-800. 5 young ladies will have anorexia. These are major challenges. Antimicrobial resistance. You know, we're becoming resistant to the antibiotics that we've got. 700,000 people are dying today, every year, now, because of AMR. In 2050, they're predicting that will be 10 million. And Jim O'Neill, who did this long report a few years ago, Colin knows Jim well, he predicted by 2050, if we don't come up with a new generation of antibiotics, it's going to cost the planet close to $100 trillion. These are major challenges. Rare diseases. There's 7,000 rare diseases. We have treatments today for only 5% of them. Altogether across the planet, there are 350 million people with a rare disease. Half of these are children. 1/3 of those children don't even reach the age of 5. We desperately need more treatment. Let me share with you a couple of anecdotes because they put into perspective how desperate parents, patients, relatives are. A couple of years ago, a lady who lives in Bath came to visit us in Oxford. And her daughter has a rare GI stromal tumor. So this tumor is about 1 in a million. So in the UK, there's probably about 60 of these individuals. And she's sitting in my office, and her daughter's physician, who lives in Cambridge, he also came. So the mother and the physician are sitting in my office, and the mother starts to get emotional. And she's getting emotional because her daughter has no hope. There is no treatment. There is no company trying to develop a treatment. What is she to do? So I said to the mother, I said, how is your daughter doing? She said, my daughter is 22 years old, and they've just removed the whole of her stomach. And I'm sitting there thinking, how on earth does any parent cope with that? You know, I have a 27-year-old daughter, and she's my life. How does any parent manage that situation? 2 years ago, I was giving a lecture in the Maths Institute on the Woodstock Road. This was on a Sunday morning. I gave this public lecture, and this lady came up to me at the end, and she said, Professor Bountra, my daughter died earlier this year with a brain tumor. If you would like to study her brain, this is where you can get it from. And she hands me a slip of paper, and on this slip of paper was her daughter's name, her date of birth, the day she died, and where her brain was being stored. This was immediately before lunch, and my wife came to pick me up. And I said to my wife, I've just had a complete stranger offer me the brain of her dead daughter. This is how desperate parents, patients, relatives, carers are. We desperately need more new drugs. We need effective drugs. Now you might think, well, why is he saying effective? Aren't all the drugs that are out there effective? Well, 3 years ago, there was a publication where they looked at treatments for solid tumors. So at the time, we had 71 treatments for solid tumors. And they looked to see how much increase in progression-free survival and increase in overall survival they provided. And the conclusion was they produced an increase in progression-free survival of 2.5 months and an increase in overall survival of 2.1 months. Of those 71 drugs, only 30 had clinically meaningful efficacy. So these drugs are out there, they're being sold, they're being given to patients. Less than half of them had clinically meaningful efficacy. We need affordable drugs. So a publication in 2014, they looked at the drugs that we had for treating cancer in the year 2000. So in the year 2000, in the UK, we had 69 drugs for treating cancer. Of those 69 drugs, 50 were cytotoxic treatments. The average duration of treatment was 181 days. The average cost of that treatment was £3,000. And in the year 2000, £3,000 equated to 20% of our GDP per capita. They did the same analysis in 2014, and now there was a further 63 drugs. The average duration of treatment was now 263 days, so gone up from 181 to 263. The average cost of that treatment was £35,000, gone up from 3,000 to 35,000, and 35,000 equated to 140% of GDP per capita in the UK, gone up from 20% to 140%. And that trend is continuing. We need more affordable drugs, and we need them quickly. For patients, even tomorrow is too late. They want those drugs today. We need more novel drugs. They need to be effective. They need to be affordable, and we need them quickly. Now, the problem we have in industry, or in the community is the way we're doing drug discovery today, it's incredibly inefficient. It is too costly, it is too risky, and it is too slow. So let me just take each of those in turn. So too costly. So in 2012, Forbes did an analysis. They looked at a number of the large pharmaceutical companies. They looked to see how much money they spent on R&D over a finite period. And they looked to see how many drugs did they launch in that period. And they divided one number by the other and came up with an average cost of a new drug. In that analysis, AstraZeneca, the average cost of a new drug was $11.5 billion. Just to put into context, to run this great university is about £1 billion a year. Their average cost for a new drug was $11.5 billion. In that analysis, Amgen, who were the best, that even they were $3.5 billion. More recent estimates of cost of a new drug, everybody recognizes is somewhere between $3 and $4 billion. It is too costly. It is unsustainable. We can't carry on like this. It's also too risky. So in 2014, there was a publication where they looked at drugs that were in development for cancer in the year 2002. So in 2002, across the world, we had 529 molecules in development for cancer. So by in development, I mean they were either in phase 1 clinical studies or phase 2 or phase 3. And they looked to see a decade later, in 2013, what happened to those 529 molecules. And the conclusion was 45 of them had made it to the market. Well, that's good. 95 were still in development, and 389 were terminated. So we, as a community, we took 389 molecules into patients and then terminated them. Just imagine how much money we spent on that. Just imagine how many people's careers went into that. And imagine how many patients were exposed to those 389 molecules. The process is very inefficient. It's too costly, it's too risky, but it's also too slow. A couple of my colleagues, 3 years ago, published a paper. They looked at when did we come up with an idea that a new target would work in cancer? And then how long did it take to take that idea into the clinic? And they concluded that it took anywhere between 6 years and 30 years. 30. 30 years to take a target from the lab into the clinic. It is too slow. Why? This industry employs some of the smartest people on the planet. Why is it so costly? Why is it so risky? Why is it so slow? I would put the reasons for this into maybe 3 broad categories. I think there are major scientific challenges. Let me go through those in a moment. There are also challenges around what I would call duplication, and I'll comment on that. And there's also challenges around reproducibility. Let's go through the science first of all. For many diseases, frankly, we haven't got a clue what causes the disease at the molecular level. We don't really understand at the molecular level what causes schizophrenia or depression or Alzheimer's in a particular patient. For many diseases, we don't have good biomarkers. So biomarkers are a way of assessing efficacy, for example, in patients. You can't say to a depressed patient, oh, take this drug, and then a week later you can't go up to them and say, oh, are you less depressed today than you were last week? Or you can't say to an Alzheimer's patient, oh, is your memory better today than it was last month? We need better biomarkers. A lot of the animal models that many of us use, personally, I think, do not predict what happens in the clinic. I do not think we will ever have an animal model that truly recapitulates all the pathology, the phenotype that we see in patients. We'll never have an animal model of schizophrenia or depression or whatever. There are also many drugs out there we don't even know how they work. Paracetamol, acetaminophen, we've all taken it. Probably 100 million people across this planet took paracetamol today. We do not know what the molecular target of paracetamol is. So if you don't know how existing drugs work, how are you going to design better ones? So these are major scientific challenges. There are also challenges around duplication. And this is something that really upsets me. There are so many academics, so many biotech companies, and so many pharma companies working on exactly the same idea in parallel, in secret, in competition. And since most of these ideas, when we take them into the clinic, 80-90% of the time they fail, you can imagine if we've got 20 companies working on the same target, they'll spend 6-7 years coming up with their own molecule, and then they take it into patients, and 9 times out of 10, they will all fail. This is a waste of money, it's a waste of people's careers, but it's a horrendous waste of patients. Ethically and morally, we believe it's wrong. There are also challenges around reproducibility. Over the past 5-6 years, there's been a number of high-profile publications from industry saying that 50, 60, 70% of the time, they cannot reproduce academic literature. We need to address this. There's no point being defensive about it. We need to sort it out. These are major challenges. Now, Julian asked me to comment on innovation, and innovation is an overused term these days. But this industry, this community, we as future patients, we desperately need more innovation. So let's just share some figures. I already said we've not had a new drug for Alzheimer's since 2002. Jim O'Neill wrote in his report, we've not had a truly novel antibiotic since 1984. For the past 4 years, 2014, 15, 16, 17, of all the drugs that have been approved by the FDA, roughly 40% per year are what we would call novel. The rest are what we call me-toos or new formulations of existing drugs. Generic drugs. If you take the top 13 pharma companies, 3 years ago, their total sales were about 300 billion, but of those, 123 billion was generic. In the year 2000, 50% of prescriptions in the US were generic drugs. In the year 2015, 86% of prescriptions in the US were generic. In 2014, Lilly, a large pharmaceutical company, 7% of their sales that year were drugs that were launched in the previous decade. 93% were older than 10 years. And in 2015, if you take the top 13 pharma companies, for 7 of those companies, their sales were lower in 2015 than they were in 2014. These are challenges for patients, for the economy. These are scientific challenges. These are organizational challenges. Let me share with you what we've been trying to do in Oxford for the past few years. And I could probably break this down into 4 things. So we've tried to pool our resources, our expertise, our infrastructure We've tried to work with many pharma companies. We've tried to get funding from many different organizations, government, philanthropy, charities, patient groups, etc. We're pooling resources to share risk. So currently, we're working with 9 large pharmaceutical companies. Each of these companies has given us about 5 million euros over a 5-year period. So we have more than 40 million euros of private funding. We're working, thanks to Lee's hard work, with 6 different patient organizations who are giving us funds and so on and so forth. The Wellcome Trust have put close to £60 million into our lab in the past decade. We're pooling resources to share risk. The second thing we're doing, we only work on completely novel ideas, novel proteins, novel protein families. We deliberately work on areas of biology that people think are impossible, intractable, undruggable. Because we believe our job in academia is to drive innovation. So we work on these new genes, on these new proteins, and we generate high-quality reagents, high-quality tools. So we purify the human protein. I'm not interested in rats and mice. We purify the human protein. We build assays for that protein. We work out the shape of that protein. Using these assays and shapes, we can design small molecule inhibitors, potential drugs of the future. We generate high-quality tools. We also generate antibodies. So by working on novel proteins and generating novel tools, we're trying to drive innovation. And these tools are high quality, not because we're clever, they're high quality because we're tapping into the resources, the infrastructure, the expertise of these 9 large pharmaceutical companies. The third thing we do, and this is probably what makes us most unique, all of these reagents, all of these tools, all of these starting points for drug discovery, we give them away to anybody in academia, anybody in biotech, and anybody in pharma. Because we believe that's the best thing we can do to accelerate science and therefore accelerate drug discovery. Now, of course, the consequence of that is you can imagine every academic who comes to see us wants to work with us because they know we're working on novel ideas, we're generating novel high-quality tools. They take these tools and they can do with them whatever they like. They can test them in models of inflammation, models of dementia, models of diabetes, models of cancer, models of rare disease, whatever. And of course, academics, all they care about largely is publication. And of course, because they've taken novel, high-quality tools for novel targets, they get high-quality publications. So we're now working with more than 300 academic labs all over the world. We don't pay these academics anything. This is a way of crowdsourcing science. By generating these high-quality tools and making them freely available, we're crowdsourcing science, so we're de-risking these new ideas, these new proteins. The fourth thing we do is that all of our reagents, all of our data, all of our knowledge, we share it with the world immediately. We don't sit on it for 12 months while we're writing up our manuscript and getting it reviewed, because in those 12 months, there'll be people out there wasting their time doing exactly what we've already done. So by releasing everything immediately, we're trying to reduce duplication and wastage. So 4 things. We're pooling resources to share risk. We're working in completely novel areas and generating novel tools to drive innovation. We're making all of these tools freely available to crowdsource science, and we release everything immediately to reduce duplication and wastage. So what impact have we had? So let me share with you just one story. In 2009, we got some more funding from the Wellcome Trust, and we said we're going to take novel proteins and we're going to generate novel inhibitors, and we're going to work with industry to generate those novel inhibitors, and then we're going to share these inhibitors with the world. Now, people said, you're mad, you'll never get industry to give you their resources to generate novel molecules, which you're then going to share with the world. Well, in 2009, GSK agreed to do that. We decided to work on some proteins called epigenetic proteins. We deliberately chose these because these were novel, people hadn't worked on them. We believe that many of these proteins will be important drug targets, not just in cancer, but in inflammation and neuropsychiatric diseases, et cetera, et cetera. So one of these families we worked on was called bromodomains. And altogether in humans, there are 61 of these proteins. And again, people said, you guys are mad. You'll never generate an inhibitor because these are impossible proteins. It's a protein-protein interaction, et cetera. Anyway, we did. We worked with GSK. We worked with a collaborator in Harvard named Jay Bradner, who's currently the head of research at Novartis. And together we generated an inhibitor for one of these bromodomain proteins. The protein is called BRD4. The name doesn't really matter. So we generated this inhibitor. It was potent, it was selective, it was cell active. And we thought, okay, well, what does it do? Now we've got this inhibitor, what does it do? Which disease is it going to work in? And at the time, when we looked in the literature, there was probably only about 2 or 3 publications on BRD4. Now, one of these publications said that when this protein BRD4 is fused with another protein called NUT, those individuals develop a rare cancer called NUT midline carcinoma. It's such a rare cancer, most oncologists can't even diagnose it. Now, Jay Bradner, working in Harvard at the Dana-Farber, had access to patients with NUT midline carcinoma. And so what he did was he took cancer cells from these patients and he put this inhibitor that we generated on those cancer cells. And he showed that, one, it stopped the cells multiplying, the cancer cells from multiplying. Well, that's exactly what you want in an anti-cancer drug. He then showed that it actually killed the cancer cells. It increased apoptosis. And when he took these patient-derived cancer cells and he put them into a mouse, the cells multiplied, the tumor got bigger and bigger, and when he dosed the compound, he showed it stopped the tumor growing. Now, this was in 2010. And we published all of this data in Nature in December 2010. So that's what, 7 years ago. It's instructive to think of what's happened in the past 7 years since that publication. That molecule that we generated, we, GSK, Jay Bradner, has given that molecule out to probably close to 2,000 labs across the planet. These 2,000 labs have taken that molecule and they've now shown that it works in a whole range of other cancers. There's data in sepsis, there's data in fibrosis, there's data in cardiac hypertrophy, there's even a publication in male contraception. We have crowdsourced science. There are now close to 1,000 publications, many of them using this molecule. When we started, there was a handful of publications. Now there's probably more than 1,000 publications. Now, based on these publications, many pharma companies and many biotech companies have started their own internal programs. And now these companies have generated proprietary molecules and they've taken them into the clinic, and we're aware of at least 20 ongoing clinical studies with proprietary molecules. So by generating this molecule and then making it freely available, we have catalyzed academic science. We have accelerated proprietary programs inside pharma. We've catalyzed translational studies and clinical studies. But on top of that, what happened is that in 2011, Jay Bradner secured $15 million of VC funding to set up a biotech around this target, and he called this Tensure Therapeutics. He set it up in Boston. This was 2011. And then in January 2016, this company, Tensure, was bought by Roche for $535 million. So we've also enabled the creation of new companies and new jobs. Now that's just one story. We've generated now more than 60 of these molecules. They're freely available. Anybody can do with them whatever they like. They're on our website, etc., etc. So what we're now doing is one of the great advantages of sitting in an academic centre like Oxford is, of course, we're surrounded by hospitals. We're surrounded by clinicians. We have access to patients, patient databases, but importantly, patient material. So what we're now doing is taking these patient samples, disease samples, cancer cells, immune cells, and then these novel inhibitors that we're generating, we're testing them on these patient-derived cells. And I think that is a much better way to come up with new targets for drug discovery. What we're also doing, thanks to Lee, is building closer, broader, deeper relations with patient organizations. Now, of course, patient organizations are very powerful in terms of telling governments where to spend their research dollars, but they're also very powerful, and increasingly so, in terms of telling payers which drugs to pay for. But we're keen to work with patient organizations because they can help us get patient material. And so we're now testing our molecules on this patient material with these patient organizations. And finally, what we're also doing is setting up a national project to focus on aging. So we're working with the University of Dundee because they've got lots of chemistry. We're working with a Catapult that's funded by the government in Manchester called the Medicines Discovery Catapult. We're working with the University of Birmingham because they've got 6 million patients, potentially, compared to Oxford with about a couple of 100,000. And we're working with the CRIC, this new institute that's been set up in London because they've got lots of novel biology. So we're bringing together these 5 centres, Dundee, Manchester, Birmingham, Oxford, and the CRIC, and we're trying to work with the whole of UK academia and industry in the UK to accelerate new treatments for aging. But the underlying premise is, even in Oxford, we do not have all the expertise, the resources, the infrastructures that we need to tackle these problems. So what are we now doing with the Oxford Martin School? What I've tried to convey is that I believe in early discovery there is too much secrecy, too much competition, too much duplication, too much wastage. What we're trying to do is bring together lots of academics, lots of clinicians, lots of pharma companies, lots of funders from charities, philanthropy, patient organizations, industry, etc., to try and work together to come up with new ideas for drug discovery. We're doing this in a much more collaborative, open, cooperative way. Now, we think this has many advantages. But what we're going to do with the Oxford Martin School is we're going to work with social scientists. We're going to work with economists. We're going to work with IP lawyers. We're going to work with ethicists. Lee is working with the European Commission. And what we're going to do is we're going to try and quantify the benefits of open research. What are the benefits for industry? What are the benefits for patients? What are the benefits for academia? What are the benefits for funders? We want to quantify all of that in a very transparent way. We want to work with patient organizations. We want to work with regulators because we need to produce data which will convince all of these stakeholders to change the way we're currently working. What we're trying to do together with the help of the school is to create a new ecosystem for drug discovery. An ecosystem which, ladies and gentlemen, we hope will help generate more novel medicines, more effective medicines, more quickly. And I hope, I pray that these treatments are also more affordable. Ladies and gentlemen, thank you very much. Lee, and that's Nick. So I should sit down and you should give these guys a hard time.
I'll do this as well. There's also questions.
Why don't I start off?
Go on, Stephen.
Hi, I'm Stephen Zimmer. I work with Chaz actually on a number of things. Obviously, Chaz, agreement on many different fronts with what you've said. But my question revolves around the issue of we know today, okay, that the biology of disease, biology in fact, and pharmacology are much more complex than the current paradigm in pharmaceutical industry believes. And I think all the things that you've said about open platform, about sharing, about lack of duplication, of trying to get at more effective and more cost-effective therapies in general are absolutely true. But is it not the case that unless we start thinking about biology and about pharmacology in a different way, we will still not move the dial, okay, in terms of how efficacious, of how, how many drugs we can generate. I mean, 90% or 95% failure rate is outrageous. It's egregious, okay? It shouldn't be. And all of these studies that have shown that, early stage research in academia doesn't get, is not reproducible by companies when they go back and look at it, egregious. But my question really is around the paradigm. The paradigm, I think, has a problem. It has failed. Okay, so the question is, in this whole discussion and all the work that you're doing, I would think that one of the elements of this has to be not only the economics, the social effects, and all the rest of it, but it also has to be about, are we doing this the right way? Okay, are we looking at this in the way that, we have the knowledge, we have the tools, we have everything we need to do this a lot better than we do. You and I are trying to do this in a better way.
That's what my wife says about me, I'm trying.
Yeah. So I think that's the question, really. It's about that.
So, Lee, Nick, do you want to go?
Oh, I think, Stephen, you got a very good point. One of the first steps for that to happen is really to break down the way we are structuring, all the, if you like, incentives for academics, which are based a lot on how many publications we do or how many patents we bring. Patents, they're easy to write. You put it in a program, made it happen, and people use that as almost a currency for the professorship. So many places. Many of them are not worth the paper they're written on, right? So by actually reassigning or reaffirming what are the main bottlenecks that we can actually address by freeing from all these restrictions that we built over the years, I think the first starting point. So we can start connecting with more people. Second thing is, of course, Chaz mentioned, this freedom to be open. We are putting data out there, which can be seen by way more than just us, the biological scientists. We can have data people looking at it. We can have other groups having experience or looking at innovation in other systems, bringing this inspiration into how to solve many of the biological problems. You see the big data coming in as well, where we actually getting help from biostatisticians to look at how this different kind of data sources and elements can be brought together so we can pool the information that is needed. Things that we never looked before, but actually using new tools, we can get there. Do you want to add?
Yeah, and I think it's also about trying to change the pathway a bit, so how you can move that open pathway into the preclinical while at the same time gathering the biology. So in a way, if you can start to develop leads, get those molecules, quantify them, generate some safety data, but also understand what's going on out there in the biology of it, so it can then be fed back into that program, that process in an open way, then I think there's a chance where you can speed things up more effectively. And then looking at multiple clinical indications, which is what Chaz has been talking about, I think is part of the solution to it. I mean, in terms of deeper structural and sort of process issues, I think there's still a lot of work to be thought about within the industry, and I think it's got to change a lot about, and you know better than I do, it's gone through various paradigms that it's looked at as being the solution to finding new drugs. I think we've actually just got to get these things safely into patients more quickly to look at the biology. And that hopefully will open up a new area, but there's still challenges with that.
Cole?
I'd like to challenge in a positive way the point which has just been made. This program has to be very supportive of industry, academia, regulators, healthcare professionals, government. Nobody wants to spend, is it £1 to £2 billion per drug taking 30 years? And the reason I make that point is that I believe what's being done here are looking for solutions. Everybody's looking for a solution to this. Currently, it's like archaeology. People are digging test pits here, they're filling them in, they're saying nothing. Somebody's coming along the next night, digging the same hole, saying nothing. Then they're digging a test pit over here. I think Professor Bountra's comment about the underlying science not being clear across a lot of medicine is a key point. I'd like to see some proper seismic survey of the whole landscape. That needs a different approach. And this is not wholly a question about intellectual property, which a lot of these problems hinge around. So it's not all about commerce. There are issues of humanity. And I think Professor Bountra made that point earlier. I read a report from the World Health Organization that 1 in 10, maybe more, medical drugs on the market in developing countries are fake. And these deal with cancer, HIV, even down to painkillers, contraceptives. So, I mean, in answer to the point you're raising about the changing of paradigm, I impel all of us here in this room, I impel all the people watching this podcast, just a little encouragement of a little more open science makes a big difference. So wherever I meet this issue, whether it's in big pharma or biotechs or in regulators, I just press people to think. It's a bit like those old things of the past. We have to put something back in the soil. This is medicine.
I think Steve's point, maybe just to add, I mean, I do worry that we don't have enough clinical pharmacologists being trained. I do worry sometimes that, I mean, I appreciate technology is making many things easier, but, you know, the complexity of what we're trying to do, I mean, you take 100 patients with Alzheimer's, they'll all be different. You know, some of them will be agitated, some of them will be aggressive, some will be depressed, some will be whatever. Even if you have identical twins, if they're living in different parts of the world, you know, they get different diseases, etc. Patients, humans, look at us, we're all completely different. We're heterogeneous. And, you know, sort of when the Human Genome Project was done in the year 2000, I, like many people, thought, my goodness, we're going to get so many great new drugs. But now sort of we realize that there's another layer of complexity on top of the genetic code. There's the epigenetics. Now we're starting to realize that in each of us, there's 1 to 2 kilograms of bacteria in our guts, and the total number of bacterial cells is more than the cells of our body. And we know how alcohol, antibiotics, diet can affect that bacterial composition. That's going to affect drug absorption and so on and so forth.
And our mental health.
Absolutely, Cole. I mean, but how do we pool all this data together? I mean, to me, I think increasingly this is becoming impossible for any company, any university, any organization, and certainly any lab. The future has to be, we need to work together, pool our resources, etc., etc. So thanks for asking that, Steve. Please, there's one here, one there.
I love what you're doing. I want to ask about an alternative that is probably equally far away from the traditional modes of discovery as yours is. So in the States, there have been 4 hospital chains just over the last couple of weeks, which have announced a new business that will go into the generics industry. Do you think that approach of private sector hospitals working together around generics could ever get to the point of, to use a word that you questioned, innovating toward new drugs?
Cole, Nick, then Lee.
Good question.
I think the answer is yes.
Yeah, I mean, I suppose it depends on where they're going to focus and what targets and how they're going to build a sustainable model around that. I think the benefits of the generics is you can get things much quicker into the market with that opportunity. Whether you're actually going to solve some of the problems and actually be able to tackle the diseases, I think is a question. So quite what the ambition of what they're trying to do, whether it's a question of depressing their costs and bringing the costs down, the question is then what you're going to do around that. So maybe they're trying to change the paradigm in the sort of payer and the sort of drug pricing area, but I don't think it's going to have much of an impact of saying, well, here's a new therapy for a new disease, unless they're going to test things in other clinical areas. So I don't know.
No, I fully agree with you, Nick, because it's almost equivalent of saying, we're now defining the diseases much more precisely, right? Think about the word cancer. The origin of the word means crab in Greek. Why? Because it is open people, and so this mass of cells which have lots of ramifications, veins, etc. That's what's a crab. So we still call the disease cancer today, but we know that different cancers have very different origins. It goes all the way down to the molecular basis of the disease. And yet we still treat as crab, right? Now, think about that. When you talk about drugs that are already existing there generically, those were designed for one or the indication. Now, if you think about how disease have to be much more targeted, I think it's a little bit, the chances of existing drugs addressing other problems are much smaller to be super efficient. But of course, for many diseases, for many populations would have diseases, that's the only thing they can get hands on. So you would try. But more and more, many disease foundations, and that's why they're working more closely with us, they too realize that this repurposing, trying to test things already out there, they're not giving very, very excellent results so far. The return has not been good. So they are now investing more and more into, let's understand what disease is, and together with groups like ours, working in the open, design something bespoke. But when they design it, they will not hold it back. They will not tend in control of one or other small group for the monopoly. They will share it. So therefore, we can actually start seeing different changes in pricing structure, access, and why not using that more bespoke, fine-tuned tool to explore biology, to understand other diseases as well.
Cole?
I think you make a very fine point, as did the previous speaker. This previous question. Science and industry move out fast. Genetics, genomics, this is a fast developing field. You've got frontline clinicians now with their stethoscope. They're not taking your pulse anymore, they're taking your physics. And it's understanding the power of these developments. And we're going back to the true physicians as it is the first form. So even though this program here seeks to find solutions, part of those solutions lie in new technology, new approaches, and encouraging new approaches. Please.
Thanks so much. I'm Desmond McNeill. I'm a professor from the University of Oslo in Norway. Thank you. Very interesting and convincing presentation. And as a political economist, I'm happy to hear you're getting some social scientists in. But I'd like to comment on the way you presented what you thought you would like them to do, because it seems to me that demonstrating or assessing the cost of this wastage actually doesn't take, you can come up with huge numbers with a large margin of error, nevertheless, to show in economic terms a massive cost. But in fact, it's so evidently clear that I would have thought it was more useful to spend your time analysing, if you like, the political economy of the situation. Why is it? I mean, there's this huge the wastage arising out of competition, huge wastage out of drugs that don't work, and yet people have the right to demand them. It seems to me that I frame it as a question: would you not be better spending your effort on the economics or political economy, looking at the rules of the game, intellectual property, the way the market works, rather than putting a number on, which in a sense is perhaps not the most important issue?
Nick?
Yeah, I mean, it's a good question, and that is something that we're looking on. So Thomas here, who's helping us around the economic bit, is really looking at the issues around the return on investment, different types of models around gaming and assets, and how you can buy assets and look at them in different ways is part of what we're looking at. And then the IP landscape as well is pretty integral. For a company to invest multi-million pounds into phase 2 and phase 3 clinical trials, there's the question of market exclusivity. So what is it going to incentivize a company to invest in that area if you say the IP framework is going to be drastically different? How would they want to do that? Would they ever do that? And for that, you've then got to demonstrate what the timescale, your probability of success. Can you increase that success rate going through the pathway and link that to a better return on investment? So the core of what we're looking at is the sort of linkage between the economics around an open versus a closed model, and then what different types of IP models you have. So you could say, well, orphan drugs shows you quite a good design where you have a 10-year exclusivity for that designation, and then that's taken through. Is there something one could explore similarly for difficult intransigent diseases where actually you do use an open innovation model that allows companies to invest and then have a period of exclusivity? We don't know the answers to that. It's a question of what the balance is at the end of it. And I think even if you went to a pharmaceutical company and you had very positive figures, you'd have to do quite a lot more to persuade them and actually try and find a pilot study or a molecule that you could take through under that type of approach and see what happened. There are other opportunities for companies to say, go back and repurpose, repatent, if you've reduced that sort of risk by taking something through in an open model into phase 2A, and then maybe they can go back preclinical and take things through on their own with a sort of closed IP system. So I think there are going to be a lot of variance, and I don't think this is going to be a one-size-fits-all. I think in different diseases you may find slightly different models are more attractive than others.
I would maybe just add sort of, I mean, if you've got complementary ideas, then please just come and work with us.
Absolutely.
Yes.
We're not pretending that we know the solution. We don't. We've just been practicing one way of working, if you like. We recognize, we feel there are some benefits, but we feel we need to quantify that and demonstrate those benefits for all the different stakeholders. We do want to look at alternative models as well, and hopefully by publishing those sorts of findings with as broad a community as possible, I mean, that's the way we're going to transform this. I mean, Julian's title to me was Transforming Drug Discovery. I mean, that's the way we're going to do it. The more of us that are talking about it, doing something about it, the more likely it will happen more quickly, etc. Cole, Lee, did you have
I
any?
just have one point on that, if I can. One of the beauties of the Oxford-Martin process is drawing in cross-disciplinary expertise. My early background is in the oil industry, nothing to do with medicine, but the scale and dimensions of the problem and the risks and the spend and the chance of discovery are the same. I think you're right to talk about political economy. Wherever we operate in the world, there are two things which most people would not know. That hydrocarbons are a national asset. They belong to the people. So governments all over the world in the political economy make oil companies, and I'm not saying they're holier than holy, but they make them operate together. They make them share. So every time you see some venture in the North Sea or somewhere in Brazil, I know some corporations mentioned, but actually that's just the front of the operator. They're made to operate in 4 or 5 partnerships, taking a share each. And that leads to sharing of data, it leads to sharing of risk, and it leads to more enterprise and less intellectual property. So I absolutely endorse what you're saying. It's a key component, and a key component to this program.
And I'd like to add just one final point, very briefly, which is actually, when you mentioned that, yes, it's so clear, that is definitely clear for industry. They understand cost, that's risk profile. But you will see that, unfortunately, for many of our academic colleagues, openness is much more of a tender topic. And you see that industry will be much more open to the openness than many of our academic colleagues.
Julian, could we just squeeze in one more question there, please? And we'll make it a quick answer.
Yeah.
Yeah,
My name is Tony Whitson. I'm a mature student at Oxford and have been for some years. I know nothing about medicine whatsoever or about medical matters, but I do know about things that impinge upon those. And that strikes me that there's a marked and distinct difference between the objectives of the pharmaceutical industry, which I regard as industrial medicine today, and those of the patient. And simply enough, I say to you that patients don't want new active ingredient drugs. What they want is peace of mind. And there's one, and that points straight towards the area which I feel is completely neglected. And that is that I have been in my professional life, I've worked with doctors. I say I'm not medically qualified, but I know hundreds of doctors and I've read all the journals and the stuff. And some years ago, in 2002, I went to Washington to attend a conference and heard a presentation, heard a talk by a professor at Kentucky University that literally made the hairs on the back of my neck stand up, because I saw so closely what he was saying, matching with what I knew to be the case in a particular condition in the United Kingdom, and I'm sure it's worldwide, but that was my own personal involvement, and I had a knowledge of 200 or 300 people who were falling into this patient field. I happen to know in the UK, a chap who lives in the UK, who's probably the world's leading research expert in this particular condition. And I'd known him for many years, and I spoke to him and said, look, I just come back from Arlington, Virginia, Washington, and this is what I've heard. And it answers, for me, dozens of fundamental questions. This guy, who is well published, very well known, frequently broadcast, and all those sort of things, said yes, he understood that. But, you know, in a sort of academic way, he didn't become involved, he didn't take note of, he didn't work on, nothing changed in that field. And what you were saying earlier on, and I think this gentleman touched on it as well, the paradigm is completely wrong. Oxford University, from my period, lengthy years I've spent here, is not joined up, and people recognise that. And I'll give you just another example.
Sorry, we need to move to a question.
1 minute, here's the question. 1 minute, and here's the question. You talked about epigenetic proteins. To me, I believe that there is a much more significant field in epigenetic traits, epigenetic behaviors, which could inform medicine in this country. Is that something that is being looked at? Of course, there's no profit in it. There's no money in it. Pharmaceutical industry not going to put billions of dollars into that. But I think you could cut short these very long times to develop drugs that work.
So maybe if I can just give a very quick answer. The reason we decided to work on epigenetic proteins is because we increasingly recognize that many diseases are massively influenced by the environment. So by environment, I mean diet, exercise, stress, sleep, early life experiences, etc. And these environmental effects are mediated via these epigenetic proteins. I suppose the other comment I would make is sort of, you know, we can't do everything. We have to focus in certain areas, and that's what we're trying to do. But thanks for asking. So, Julian.
Okay, I can see that there are other hands still up, and maybe one or two of these guys can be mugged afterwards. But before we thank them, Lars, I would like to draw your attention to a couple of other events here at the school that I think may be of interest to you. This is, of course, one of the events we have in our series on health, New Perspectives. Next week, at exactly the same time, we have Professor Kevin Marsh, Professor of Tropical Medicine, talking about prospects for new tools to control malaria. He's been working for many years on this in East Africa, chair of the World Health Organization's Malaria Policy Advisory Committee, so a world expert in that field, and promises to be another great talk. The following week, picking up on some of the things that were talked about today, antibiotics, to regulate or not, and that is a joint talk. Professor Stephen Hoffman, who's an international lawyer, specializes in global health law, and Professor Julian Savulescu, who is the Uhiro Chair in Practical Ethics. So a philosopher and a lawyer addressing that. So I hope that will appeal. And another event just to draw your attention to, next Wednesday here at 5 o'clock, we have Dr. Shengen Fan, who is Director General of the highly influential Washington think tank, the International Food Policy Research Institute, talking on innovative food systems. That's just a quick run-up of the next couple of weeks here in this room, but I would like to thank, all 4 of the people who stood up at the end. I mean, we're very excited at the school to be working this program, but in particular to thank Chaz for a passionate talk and actually a bit of a masterclass in the art of public speaking, if nothing else. So a really warm thank you to Chaz. Thank you.